give you a brief overview of some of that and take you through a little bit, but in the lab, we're going to be looking at a specific process that has a sugar that has a dye already attached to it. That same one you actually used, and it was separate from the sugar in the glucose study when you used the UV vis, remember? And you were looking at how glucose was being broken down. And so this one is actually already attached to the sugar, and you guys will be observing that by UV vis, and I think also by NMR. I'll have to double check the experiment. So, HIV. This slide actually really hits home to me because I worked on one of the anti-HIV compounds that inhibited HIV protease when I was in my graduate work, and so I got to do an internship with Abbott Laboratories in Chicago, and they essentially wanted to try and develop, or not develop, but try and redo the synthesis and streamline it so that it was essentially more cost effective, and so hopefully that cost could then be passed down to the consumer. They were generating trainload cars of waste for a specific subset of synthesis steps, and they wanted to streamline that. So they've been targeting HIV protease for actually quite some time. The one on the right, which is sequinavir, that targets HIV protease, and you can see the crystal structure of that over there, and that is co-crystallized with sequinavir. And the two compounds that I was working with were actually peptides, and they were very similar to sequinavir, and those were lopinavir and ritonavir. So lopinavir and ritonavir was a combination therapy developed by Abbott for disrupting HIV protease. HIV protease can actually mutate, and it can get around the drug. And so they were developing that as a combination therapy, so when and if the protease enzyme mutated, that they could inhibit the mutated form with the other molecule. So you had a combination of two molecules in there, and that took care of both the non-mutated form and the mutated form of that enzyme. So really cool way to target the enzyme. So the nevirapine that's bound to the HIV is bound to the reverse transcriptase enzyme for this. And we're going to talk a little bit about what happens when small molecules bind to enzymes, what happens to the enzyme, what happens to the small molecules, and even when two substrates come in and bind to the enzyme, what that looks like and what that entails. And that model's changed over the years, and we'll take a look at proof of how that has changed. Does anybody have any questions on that slide? What do you think they have to do to get these crystal structures of these enzymes? What do you think they have to do? Dehydrate it? Maybe. Yeah, I'm talking about crystallography, like how?
Do you mean like how do they introduce this into a human system without a...
No. How do they determine the structure for like the reverse transcriptase or the HIV protease? How do they do this? Do you think it's easy? Oh no, it is definitely not an easy process.
I know you mentioned early on about the beginning that they did a wet crystallography. Can they do that with this?
I don't know the answer to that. I'm assuming that there are some crystal structures that will have water in their form, and that those are easily identifiable by X-ray crystallography. I know that for a fact. But the conditions, really developing the conditions to actually crystallize these and getting enough of these enzymes without changing the enzyme, but also really just getting enough of the enzyme to work with, right? Because you're not going to be, you're not going to be synthesizing that enzyme. It's huge, it's complex. I could be wrong. Maybe there have been some that have been synthesized in order to study, but I.
Thinking synthesize, I was thinking like, how would you, if you put it in crystalline form, take out the water, you would dare to change the structure because the structure is determined by the residues and the interaction with water.
And it is true.
So I was thinking like, how would you maintain the structure and still do crystal water?
Right, but you also have to consider if they already know that it's an inhibitor, then they're co-crystallizing it together, so they assume that that is the state that it's in if it's able to co-crystallize together. That they assume that that is the active state of the enzyme. Yep.
I have a question on that slide.
Yeah.
So now that the viral DNA is integrated in the host DNA, does that mean that cell codes for HIV virus?
I would say yes. Yeah, absolutely.
Do humans do that?
Where it hijacks the cells?
Yeah.
Oh yeah, that's how it works. Yeah, that's exactly how it works. It hijacks the cells and turns those cells into factories, essentially, to replicate the virus. So, yeah, COVID-19 does the same thing. Yeah, and it again, of course, it's a different mechanism, but yeah, viruses are really weird, nasty things. Enzymes. Enzyme classifications, there are 6 major classifications of enzymes. I want you to know all 6 of these and what they do. Okay. Oxidoreductases, these are redox enzymes. That is simply just shuttling electrons, okay? In this case, you're looking at proton shuttling for NAD and NADH between ethanol and acetaldehyde. And you can see that ethanol gets oxidized to acetaldehyde. Acetaldehyde is actually one of those molecules that's responsible for hangovers and tends to proliferate and stay in your system for a little while until it gets oxidized up to acetic acid. Transferases transfers one functional group from a molecule to another, or puts on a functional group, and in this case you can see that you are transferring one phosphate from ATP over to D-glucose to make D-glucose 6-phosphate, and therefore liberating ADP, which is adenosine diphosphate. So you have the phosphate group transferring over during phosphorylation. Hydrolases, this is hydrolysis, okay? So the peptide here is being hydrolyzed, and you can see you now have a shortened polypeptide on the other side with a amino acid residue that's getting popped off there. I think that's supposed to be lysases, but it says lyases. Anybody think that's typo?
Well, it could be like the plural form, like lysase,
I'm not sure.
I got a feeling that was
right?
You think it's right?
Yeah, that's right.
It is? Alright. Because I'm thinking like to lyse,
No, lyse is the action, but lyase is a classification.
but lyase is
Yeah.
a classification. Okay, interesting. Because it's catalyzing the removal of a group, right? So I'm considering it like lysine, right? So, but it's lyase.
Well, any enzyme has to have the
Oh, ACE,
ending,
the ACE
so-
ending, that's correct. Thank you, I appreciate that. Yep, that's true. Okay, isomerases, this is isomerizing, okay? Isomerizing bonds. In this case, malate to fumarate, this is what type of an isomerization? Organic chemistry, what is it?
It went from cis to trans.
It is a cis-trans isomerization. ligases, this is doing what?
It unzips your DNA.
It's splitting things apart. It's cutting things apart, right? All right. Any questions on any of those 6?
Would you say, liases do again?
Catalyze removal what?
So the one that they give an example of is decarboxylation. So again, I am assuming that liases are cutting pieces off. They're knocking off functional groups or splitting molecules. No? Am I wrong?
An enzyme that catalyzes the joining of specified molecules or groups by a double bond.
Cause it's adding
Mm.
the CO2.
But that's not what they're showing here. They're showing it taking off a carboxylate.
Oh, wait, something else says that it catalyzes the breaking of various chemical bonds.
Ah, see, I was
Yeah.
right. There it is.
Oh, why?
So I'm assuming there are several different subtypes of liases that do different things, but again, lysing or splitting, I think that's where the name comes from, where you are cutting pieces off of it. Okay. So let's talk about kinetics. Reaction kinetics, we're talking about how fast a reaction proceeds. How fast a particular reaction proceeds. How do we speed up reactions? If you're running a reaction in organic chemistry, how do you speed it up typically?
Increase heat.
Oh, I heard a few different things. Okay, so Caleb says increase heat. Will that work?
In some cases.
In some cases, that's true, yeah.
Concentration.
Concentration is another one, right? So increasing concentration, what else? Adding catalyst, which is essentially what we're getting ready to talk about, okay? How does concentration work?
It's more available molecules, but like these shot layers,
They find.
In an equilibrium process, it can be Le Chatelier's principle, right? But it's a lot about molecular collisions, right? Effective molecular collisions. So when you're increasing the concentration, then you have more molecules that are in solution that can find each other and have more effective collisions as a result of that, okay? This is a graph showing you essentially the dependence of linear concentration from a single substrate. And we're talking about first order kinetics here. Whereas time goes by, the substrate decreases in concentration. Can you give me an example of where this might take place? Where might this take place? A single substrate decreasing in concentration over time.
You eat something, over time this happens, as your body breaks down sugar.
Okay, so if something's breaking down, what kind of reaction classification is that?
Analysis?
Decomposition. Decomposition.
We're really going back there.
So a process of decomposition would be a first order type of situation. Take the logarithmic scale of both of these, and this sets you up for y equals, good old y equals mx plus b. Let's take a look at that. Okay. If you put this in y equals mx plus b form, then, and it's not right now, okay, then your slope is equal to what? Negative k. What is k? Nope. hint, it's something that has to be determined experimentally.
It's not temperature, is it? The rate of the reaction?
That's right, it's the rate constant for the reaction. Yeah, yep. So, setting this up, and running that, determining whether or not you have first order kinetics, right? All of this plays a role in determining that rate constant, okay? And rate constants, and this is important, rate constants can only be determined experimentally, okay? That is actually how mechanisms in organic synthesis are actually derived. Mechanisms are actually derived from kinetic studies, reaction kinetic studies. It's not the other way around. It doesn't actually feed into it. There may be a theoretical mechanism that somebody proposes, but until reaction kinetics actually uphold that, that notion or that particular mechanism, then it hasn't essentially been proven yet. There's actually mechanisms and I mean some very common drugs where we don't know exactly how they work. Tylenol, acetaminophen is one of them. We actually do not know everything about how Tylenol works. And that's why the dosage is the way it is. I mean, you can actually overdose easier on Tylenol than you can for ibuprofen. So you have to be more careful with that. But we still do not know all of the pharmacology associated with acetaminophen. So the observed rate of a reaction depends upon these four, which we just talked about: the order of the reaction, whether it's first, second, third order, the concentration of the reactants and the products, the temperature, and value of the rate constant, which must be determined experimentally. Why do we use the microwave sometimes for doing chemical reactions?
It excites water molecules.
It excites water molecules.
Well, it can also keep it at a more consistent temperature than if you were to do it.
That is actually true. You have a probe that goes in it. You can actually monitor the temperature very, very well on that. What else?
The molecules move faster, so they find each other faster.
But that is a factor of what?
The heat.
As a factor of the heat. So for a while, in organic synthesis, when they started using microwaves to do chemical reactions, there were people basically reporting a microwave effect on the reaction. And people were arguing about this back and forth, like, is there a microwave effect, is there not a microwave effect, and how is this actually coming to fruition? And I think there are still some arguments, but most people will agree that that is not the case. What ends up happening is you have sealed vessels, unlike you running reactions in flasks on your bench. So what can you achieve? Pressure, which then allows you to actually superheat the solvents above their boiling points, and therefore you are increasing the temperature higher than you normally could in an open round bottom flask, because why? You would lose all the solvent out of there and your reaction would go dry. So this is how the microwave actually has an advantage over normal open round bottom type containers.
So then would pressure also be a factor
Mm.
on this side, or is it not?
It's only typically a factor for something else. Gaseous reactants. If you have gaseous reactants and you increase the pressure, what are you doing? You're increasing the concentration through solubility. And as a result of an increase in pressure and an increase in concentration, then you are increasing the reaction rate based upon the concentration. This is a really good review for you guys. It's good. So, thermodynamics. We have to consider, is that reaction even favorable under specific or given conditions? And so determining what that looks like, whether or not you have a positive delta G or a negative delta G, and this is essentially just showing you that this particular initial state, final state is a favorable process where you now have a delta G naught, and your final state of your products is of lower free energy, and therefore the reaction should be spontaneous under those specific conditions. What we typically see is reaction coordinate diagrams. You guys should be very familiar with these. This specific type would be one of the more simpler ones.
We have 20 minutes left of class.
What?
Yeah.
Oh no.
27.
Okay, all right, okay, done. See, I'm getting out of control. All right.
I was taking good notes.